is Beverly Baker, Account Coordinator at IDDI, and I would like to welcome you to our third webinar on biomarker-based clinical trial designs. We're happy to welcome 120 attendees today. Just a quick overview of IDDI. As most of you know, since 1991 we provide biostatistical and e-clinical services in phase 1 to 4 clinical trials. These include trial design, biomarkers validation, data management, statistical analysis, medical writing, and supporting contacts with the FDA and EMA. We have developed our own web systems. We have an interactive web response system for patient randomization and treatment allocation, and also internet coding of medical terms. Over the past 20 years, we have developed a thorough experience in oncology, cardiology, ophthalmology, and also biosimilars. So the content of today's webinar will focus on biomarker-based designs of clinical trials. We will review the recently proposed designs, including adaptive designs, and discuss the main issues related to their application. We will also analyze the ability to validate the biomarker and/or to establish the effect of an experimental therapy in patient populations defined by the presence or the absence of the biomarker. We will consider biomarkers that modify the prognosis of individual patients, or prognostic biomarkers, and biomarkers that predict how individual patients will respond to specific treatments, predictive biomarkers, which are also called effect modifiers. All the discussed methods and concepts will be illustrated using real-life examples from oncology. After the webinar, you will see a survey form which we would appreciate you completing. It's a very small form, and on this form you will also have the opportunity of asking further questions. And we will address these questions afterwards and get back to you with the answers, as well as a copy of the presentation in a follow-up email. So, who are our speakers today? Well, initially we were having Dr. Stefan Michiels and Dr. Mark Boyce. Stefan, unfortunately, is stuck in Frankfurt, snowbound, and desperately trying to get back to Brussels. So Mark has taken over the prospect of talking to you for about an hour. Dr. Mark Boyce is chairman of IDDI, based in Louvain-la-Neuve, and associate professor of biostatistics at the Hasselt University in Belgium. So I'll pass you over now to Mark, and I hope you'll enjoy the webinar.
Good afternoon, or good morning for those of you who call from the US. We will discuss indeed biomarkers today, and as a way of introduction, let me first show a slide that shows the ideal situation where we would like to be, where indeed a patient population can be subdivided into patients who respond to a certain therapy and who have no toxicity from that therapy versus another group of patients in whom the treatment doesn't work and or is toxic. This is the ideal, and of course we are not there yet, and before we can get there we need to validate the biomarkers upon which this division of the population will be made. So before we start our discussions of trial designs, let me define biomarkers as stated on this slide and the different types of biomarkers that we will and will not discuss. The two types of biomarkers we will discuss today are called prognostic or predictive. The prognostic biomarkers have an influence on the outcome of patients regardless of treatment, whereas the predictive biomarkers have an influence on the effect of specific treatments on the clinical endpoint of interest. We will not discuss surrogate biomarkers, which in fact later might replace a clinical endpoint, but this is still science fiction at this time. So the types of biomarkers that we will discuss today are essentially those that are measured once before treatment, in other words at baseline, before a patient is entered in a clinical trial. And again, these biomarkers can be prognostic or predictive. So it's going to be of interest to us to look at two different effects in a trial. The effect of a treatment, and the effect of this treatment may be very well known if this is a treatment that has been used for a period of time already, or it could be unknown if this is an experimental treatment still to be approved for use in the population of interest. And then we have the effect of the biomarker, which itself can be unknown if the biomarker hasn't been validated yet, or can be known if this biomarker has been validated already. And so there is a gradation or a continuum of evidence for both the effect of treatment and the effect of the biomarker, and we will discuss different situations where these effects are known or unknown. So it will be useful in our discussion to distinguish between the two trial phases of interest, phase 2 or phase 3, whether the treatment effect is known or not, whether the biomarker is prognostic or predictive, and whether the biomarker itself is well known already, is potential or putative, or is completely unknown. And so for different combinations of these different characteristics of trials, we will look at trial designs that can be used, and we will illustrate these trial designs with actual examples. So Stefan, who's now hopefully still in Frankfurt, bound by the snow, was supposed to talk about retrospective analyses and prognostic biomarkers. So I'm going to cover his part of the talk and begin with prognostic biomarkers and give you the example of gene signatures. So one of these gene signatures in breast cancer is called MammaPrint. This signature was in fact developed based on 78 patients, and the signature was based on whether the patients had or did not have distant metastases within 5 years. So the idea of the signature was to divide the population of patients into those who were at high risk of distant metastases and those who were at low risk of distant metastases. And this was based actually on the gene expression profile of these patients. So eventually the method developed in Amsterdam identified 70 genes with the highest correlation with distant metastases, and these 70 genes formed the signature. So the gene signature was identified retrospectively and was then prospectively validated with retrospective series of patients. So the validation was prospective based on retrospective data. So we're not talking about prospective trial designs yet. The first validation study was performed in Amsterdam on 295 breast cancers from that center, and this was criticized because the validation series included 61 patients who had also been included in the training set. Be that as it may, the accuracy of the signature was found to be 93% sensitivity and 53% specificity. This was a good result, but it had to be validated from an outside source. And so a second validation study was performed with 307 patients from 5 European centers, and in that second validation study, the sensitivity of the signature was found to be 90%, slightly lower than in the initial validation series, and the specificity was 42%, again, slightly lower than in the initial validation study. So these were retrospective validations. Likewise, if we look at predictive biomarkers who had to be identified, most of the biomarkers we have today came from retrospective analyses of randomized phase 3 trials. For example, KRAS mutation in colorectal cancer or EGFR mutations in non-small cell lung cancer. So turning to the KRAS mutation, in fact, it was seen early on in the development of treatments for colorectal cancer that patients who had KRAS mutation did not respond to certain treatments, and as you see here, there were almost no responses in the KRAS mutated patients, and there were responses in the KRAS wild-type patients. So this seemed to indicate that KRAS mutation was a predictive factor for the effect of these treatments. More importantly, when a trial was performed on all patients of panitumumab versus best supportive care, it was indeed confirmed that patients who had a KRAS mutation did not seem to benefit from panitumumab, whereas patients with KRAS wild-type tumors seemed to enjoy a large benefit from that treatment, from panitumumab, and therefore it was confirmed that KRAS mutation was indeed a predictive factor here, or a predictive biomarker. The p-value of the interaction test in this trial was highly significant, confirming the predictive value of KRAS mutation. But this again was a retrospective analysis of a randomized phase 3 trial. How can we use biomarkers prospectively to validate their benefit or their use in clinical practice? And this is often done through clinical utility trials, of which I'm going to discuss two: MINDACT and TAILOR-X in early breast cancer. So these are now phase 3 prospective trials where the treatment of use is known. In fact, it's chemotherapy for early breast cancer. And in fact, the idea of these trials is to validate the prognostic value of the signatures, MammaPrint in one case, and Oncotype DX in the other. So the first type of design, which was used in the MINDAC trial, was to actually look at the biomarker, in this case the MammaPrint signature, and the clinical risk of the patient, and this is done using adjuvant online. And then patients who are low risk by both the biomarker and the clinical risk are excluded from the trial. Patients who have a concordant high risk by both the signature and the clinical risk are excluded from the trial, and it's only those patients in whom the biomarker and the clinical risk differ that are randomized between standard treatment or experimental, and here what we mean by standard and experimental is in fact chemotherapy or no chemotherapy. And the purpose of the trial is to find out if patients who are at low risk by the genomic signature can be preserved from receiving a chemotherapy that they do not need. So this is the full trial design. It's a little more complex, but essentially the same as the design I've just shown here. So this design is a simplified version of the MINDAC actual design, but the main idea again is to randomize patients in whom the clinical pathological features of the patients are discordant with the 70-gene MammaPrint signature. The other trial design that I wanted to mention here with respect to prognostic factors is the intermediate risk randomized design, and this was used in the TAILOR-X trial, where the biomarker now is Oncotype DX. Patients who are at low risk by Oncotype DX are excluded. Patients who are, and they will not receive chemo. Patients who are at high risk by Oncotype DX are excluded and they will all receive chemo. And only patients who have an intermediate score by Oncotype DX will be randomized between chemo and no chemo. So again, this is the full TAILOR-X design. It's essentially the same as the one I just showed. This trial, we randomized a large number of patients to prospectively validate the interest of the prognostic biomarker, in this case Oncotype DX. So far for prognostic validation or prognostic biomarker validation. What are some of the issues with the clinical utility trials? First of all, very large sample sizes are required. As I showed, Oncotype DX will have a sample size of about 10,000 patients, and the other trial, the MINDAC trial, will have a sample size of about 4,500 trials. So these are large trials that will require a long duration of follow-up. It's a bit unclear statistically exactly what hypothesis is being tested, because it's well known that chemotherapy has a benefit in these patients. It's just the magnitude of the effect that will drive the decision to give chemo to these patients or not. And then finally, prognostic biomarkers are generally less useful to choose a treatment than predictive biomarkers. So the remainder of this talk will be only focusing on predictive biomarkers. Again, these are biomarkers that will predict whether a treatment has an effect or does not have an effect on some subsets of patients. So let's turn now with prospective trials where in fact we know the treatment effect, but we do not know if the biomarker has a benefit. And I'm going to discuss two designs that are appropriate in phase 2. So again, the effect of treatment is known, but the benefit of using a predictive biomarker is in fact not known. So the two trial designs that will be covered here are so-called crossover designs, and I will show you an example of molecular profiling, and the Bayesian adaptive design, and I will show the example of the BATTLE trial in non-small cell lung cancer. Turning to the crossover design first, this was a trial of molecular profiling that was presented by Dan Van Hoff at the AACR in 2009 and has just been published in the Journal of Clinical Oncology a couple of months ago. The idea of this trial is to take patients who've received several lines of treatment for advanced disease, and this is a mixed bag of advanced disease. It's not specific to a certain tumor type, and for whom no further therapy is available. These patients have a time to progression on their last line of treatment, and then molecular profiling is implemented on these patients. A tumor biopsy is taken, and then by IHC, FISH, or microarray, a certain target is identified to try and choose a treatment that is appropriate for these patients. So the second line of treatment that these patients will receive in the trial is the targeted therapy, and the target is selected based on the molecular profiling. The idea of this design will be to compare the time to progression of these patients on the last line of treatment versus the time to progression of these patients on the targeted line of treatment. So here is the schema of the trial. The patient is consented, the eligibility is verified, a tumor biopsy is taken, the molecular profiling is implemented, and then if a target is found, then the patient is treated accordingly. If a target is not found, then the patient is treated according to the clinician's choice. The essential idea of this trial is to define the time to progression ratio, which is again the duration until progression on the targeted therapy divided by the duration of time to progression on the last line of therapy before the targeted therapy. And it's well known that for most advanced tumors, in fact, TTPR becomes smaller and smaller. When patients receive successive lines of treatment, their time to progression tends to become smaller and smaller. So in general, we would expect the time to progression ratio to be less than 1. This trial was designed to test the hypothesis statistically that at least 15% of the patients have a time to progression ratio in excess of 1.3, which is something that you would not expect given the natural history of most tumors. Here are the results of this trial. The proportion of patients who had indeed a time to progression ratio in excess of 1.3, that means a longer time to progression with the targeted therapy than with their last line of treatment, that proportion was 18 out of 66 patients. So it's 27%, and that proportion was comfortably larger than the 15% that had been hypothesized in the trial protocol. If we look at this proportion by tumor type, you see that it was in fact higher for some tumor types, such as breast and colorectal, than for others. And interestingly, among the 18 patients who indeed had a time to progression ratio in excess of 1.3, none in fact would have received the same drug if the physician had had a choice. The patients would have received some other drugs. Now, what were the drugs that showed a time to progression ratio in excess of 1.3? Here is a list of these drugs. As you'll see, most of these drugs are approved drugs that are used, but not particularly in the conditions that the patients had. And so these choices of drug, again, are driven by the molecular profiling, not by the tumor type. So this was, of course, a very interesting idea to try to target the tumor with the appropriate treatment based on molecular profiling. There were some limitations in this design. The first limitation was that only 2/3 of the patients could have the molecular profiling. So this, I suppose, is unavoidable with such techniques. More importantly, this was a non-randomized trial. And so even though the results seem impressive, given the hypotheses that were tested, there is in fact no evidence that the molecular profiling gave better results than the physician's choice would have. And so really this trial should have been randomized between molecular profiling versus best choice given the investigator choice. And that would have provided a more convincing evidence that indeed the molecular profiling helped. Another question, of course, is whether TTPR greater than 1.3, the time to progression ratio, is that a relevant endpoint? And finally, statistically, this crossover design, where patients undergo 2 lines of treatment, one untargeted and the next one targeted, this design is actually inefficient if there is a low correlation between time to progressions in first line and second line treatment, or in this case in the last line of treatment and the targeted line of treatment. So this has to be remembered that if there is little or no correlation between successive lines of treatment, this design in fact is not efficient. But given these limitations, this was a very interesting idea. Another interesting idea for phase 2 trials of biomarker-based choices of therapy is the so-called Bayesian adaptive phase 2 design, where we have putative biomarkers, and in fact here we don't have a single biomarker, but we have a panel of potential biomarkers. And so based on this panel, we can define different profiles. So here 1, 2, 3, 4, etc. profiles, which in fact will drive the choice of treatment. So patients who have the molecular profile number 1 or the biomarker profile number 1 will in fact be more likely to receive treatment 1 but they may also receive treatment 2, treatment 3, treatment 4 in a randomized fashion. So here, in fact, is the allocation ratio between the various treatments that is based on the profile, on the biomarker profile. And in fact, not only is the treatment probability chosen by the profile, but it's adapted as the trial proceeds, and it's adapted given the results of the patients already entered in the trial. So the various probabilities of giving treatment 1, 2, 3, 4, etc. are based not just on the profiles initially, but also on the results of the trial itself. That's why it's called an adaptive design. And the reason it's called a Bayesian adaptive is that Bayesian approach is used to calculate the probabilities of giving these various treatments. And these probabilities are updated as the trial proceeds. So one example of such a trial is the BATTLE trial in lung cancer. Another example is the I-SPY2 trial in breast cancer. But these trials essentially use the same approach, and so I will describe the BATTLE trial in more detail here. So the idea of this trial is to evaluate targeted therapy agents with different biomarker profiles in stage 4 recurrent non-small cell lung cancer. The endpoint of interest here is 8-week disease-free survival rate. So the characteristics of the BATTLE design is that we want to treat more patients in the more promising groups according to the patient's biomarker profile. We start with equal randomization. In other words, initially the patients are randomized equally between the treatment groups, but after 20 patients are entered in each treatment group, then adaptive randomization kicks in, and the allocation ratio between the different treatments is modified depending on the results seen in the trial. We want to early stop, for lack of efficacy, the biomarker by treatment group that show too little efficacy. We would like to provide an accurate estimate of the true disease-free survival in each of the biomarker by treatment groups. And the idea of the design is to borrow strength from patients treated with the same agent or the same treatment, but who have a different biomarker profile. Now, obviously, these are all very valid aims, but it's perhaps a lot to ask from a single trial to do all of this simultaneously. At any rate, here is the BATTLE design in more detail. Here you see the biomarker profiles, EGFR, KRAS and BRAF, VGF, VGF receptor, RXR and cyclin D1. These were the biomarkers analyzed, and here are the different biomarkers profiled here, depending on whether these different biomarkers were positive or negative. And so you see that initially the frequency of these biomarkers that was assumed is indicated here on the right-hand side of this slide, and the different treatments under investigated are listed at the bottom here: erlotinib, sorafenib, vandetanib, and erlotinib plus bexarotene. So these different treatments would be chosen according to the biomarker profile of each patient. Now there are some issues with this design, very attractive as it may seem, and the issues are listed here. First of all, we need to have some prior information to form and rank the biomarker groups. In other words, we must have some idea, at least preliminary ideas, about these biomarkers. We must know something about these biomarkers to be able to integrate them in this design. The second thing we need before this trial starts is a prior distribution for the treatment effects. And again, these treatments were known, at least to some extent, and therefore we had some idea, or the authors of this trial had some idea about the treatment effect in the various biomarker subgroups. A third choice to be made in this adaptive design is when to start the adaptive randomization and what allocation ratio to choose, how to imbalance the randomization depending on the results seen in the trial. Another big issue with these designs is the adaptive randomization. Is it more ethical to adaptively randomize the patients, or is it more ethical to randomize the patients equally between the different treatment options, given that we're not sure what treatment option is best? This is a big issue that I think needs a lot more careful thought. We need also to have a fast outcome assessment, and that is why the authors chose here the 8-week survival, a disease-free survival or progression-free survival. That is a fast outcome to measure. Obviously, these designs are very impractical if the outcome takes a long time to be observed, such as PFS or even overall survival. So these designs are particularly suited where you have a fast outcome, such as here, the 8-week progression-free survival, or the I-SPY design, which looks at pathological complete response rate in the neoadjuvant setting. These are outcomes that are measured early and therefore are suitable for these adaptive designs. And finally, the same problem here occurred as in the previous trial, that of course the biomarker profile may not always be known for all the patients, so this obviously is unavoidable. What about phase 3 trials? If we want to validate a predictive biomarker in the phase 3 setting, and assume now that again we're not so much interested in the effective treatment as we are in finding or confirming the predictive value of a biomarker. So the 3 designs we can use in this situation are called the RANDOMIZE-ALL design, the interaction design, and the biomarker strategy design. And I'm going to turn to these 3 examples in turn. The RANDOMIZE-ALL design is quite a simple design. Patients are in fact randomized between standard and experimental, and then the biomarker value is determined in all patients. An example of such a trial is the EORTC so-called P53 trial in advanced breast cancer. The hypothesis here was the benefit of taxanes would be larger in P53 positive patients. So the idea was to look at the design as testing both an overall question about the benefit of taxane, but also looking at whether the benefit of taxane was in fact larger in the P53 subset. And the P53 subset was assumed to be about 1/3 of all the patients. So the trial was designed to have 80% power of detecting a hazard ratio of 1.25 at this significance level, and the trial was also planned to test for the interaction between the effective treatment, in this case the effective taxanes, and the P53 positivity. The trial is still ongoing and the results are not yet known. What are the problems with such trials? Well, the main problem with RANDOMIZE-ALL design is that it's not efficient if these conditions apply. If there is a strong rationale for believing that there will be no effect in the biomarker negative subset, in this case in the P53 negative patients, then of course it's not a good idea to do a RANDOMIZE-ALL design. It's better to do a targeted design where P53 plus patients are entered and not P53 minus patients. If the prevalence of the biomarker positive is very low, again these RANDOMIZE-ALL designs may have very little power. And finally, we have to worry about the predictive value of the assay and make sure that the assay is quite predictive, that the positive predictive value of the assay is high enough for the positive biomarker subset to be identified reliably. Again, the biomarker may not be obtained for all patients, and this is going to be a limitation of all our designs. Another design, in fact, is quite similar but slightly different. It's called the interaction or the biomarker-stratified design. The difference here between this design and the RANDOMIZE-ALL design is that here the biomarker positivity is in fact assessed prior to randomization, which by the way has the benefit that patients in whom the biomarker cannot be evaluated will actually be excluded from the trial. So this trial only includes by definition patients in whom the biomarker value is known. And that might of course be a biased subset, but it's a subset in whom the hypothesis of interest can be tested. And so after the biomarker value is assessed, the patients are then randomized between standard and experimental. An example of such a trial is called the MARVEL trial in non-small cell lung cancer. And here is the flowchart of this trial. 1,200 patients with tumor tissue were in fact randomized, depending on the value of the FISH test. So these patients were tested for EGFR mutation by a FISH test. Patients who were FISH positive were randomized between alatinib and pemetrexed, and patients who were FISH negative were also randomized between alatinib and pemetrexed. So all the patients were randomized between the same treatments, but they were stratified by the biomarker positivity. Again, this trial is ongoing. Now, issues with interaction designs are that oftentimes the biomarker is not known or poorly known before the trial starts. So it's difficult to stratify patients based on a biomarker that is still in development. In such cases, the RANDOMIZE-ALL design may be a better option because it's always possible to determine the biomarker value retrospectively in a RANDOMIZE-ALL design. And again, just as before, the power of the interaction test is quite low, and therefore relatively large sample sizes will be required, or a sensitive endpoint will have to be used for these trials to have a good chance of finding an interaction. Now, very often, endpoints used in these trials are progression-free survival or even overall survival. The problem is that these are not sensitive endpoints. So if an interaction design is considered, it's probably a better idea to choose a more sensitive endpoint, such as PSA in prostate cancer, or even simply tumor measurements in solid tumors, so that the tumor measurements over time can be used as an endpoint instead of, or in addition to, progression-free survival. And finally, the last class of designs in this particular set is the biomarker-based strategy design. These biomarker strategy designs come in two versions. The first version, in fact, is a randomized design between a biomarker-based strategy and a non-biomarker-based strategy in which all patients receive the same treatment. So in this design, all the patients who are randomized to receive the standard treatment will receive that treatment, whereas in the biomarker strategy, only the patients who are biomarker negative would receive that treatment, and the other patients would receive some experimental treatment, the effect of which is thought to depend on the positivity of the biomarker. An example of this trial was a Spanish trial called the EORTC-1 trial, where patients randomized 1:2, so the randomization ratio favored, in fact, the biomarker-based strategy arm. The control group received docetaxel cisplatin, so all the patients here received the same treatment, whereas in this group, the biomarker-based strategy design, patients received the treatment according to their level of EORTC messenger RNA. So patients who had a low genotypic group were randomized to receive docetaxel cisplatin, just as in the control group, whereas patients who were in the high genotypic group received an experimental treatment consisting of gemcitabine docetaxel. So cisplatin was replaced by gemcitabine in these patients. Now, the problem with this design, and it's a very obvious problem, is that if, in fact, the effect of experimental treatment applies to all patients, regardless of the biomarker, then, in fact, you will not see this in this design, because the patients in the biomarker strategy design, some of them at least, will receive the better treatment, and you have no similar treatment here in the patients randomized to control. So there is confounding between the predictive effect of the biomarker and the treatment effect of the new therapy. Again, this design has very low statistical power, and so you need to talk about very large sample sizes here before you can test the hypothesis of interest. A modified biomarker strategy design addresses the second of the problem, which is the confounding between the predictive value of the biomarker and the effect of the new experimental treatment. So here, we randomize patients again between the biomarker-based strategy, but the control arm here is also randomized. It's not based on the biomarker, but this is a randomized arm between standard treatment and experimental treatment, such that the comparison now between the biomarker-based strategy and the non-biomarker-based strategy is a fair comparison. Now, it's obviously only a fair comparison if the same proportion of patients receive standard in this group as in that group, and therefore the randomization ratio must be adjusted here to reflect the prevalence of the biomarker-positive subset. So this is a limitation of this design. The randomization ratio must reflect the biomarker prevalence, which is often unknown in advance. And so we must have a method to adjust the randomization ratio or the allocation ratio as the trial proceeds. Another problem, but this is, I guess, common to all the designs that we've discussed so far, is again the very low statistical power of this design. And here it's obvious that, in fact, the random strategy will lead to the correct treatment, even though we don't determine the biomarker in these patients, but half of them, just by chance, will receive the correct treatment randomly. And so this design will in fact dilute the benefit of the biomarker-based strategy by the patients who receive the correct treatment in the control group. So all of these designs have in fact in common the very low statistical power and the large numbers required. So here Stéphane was supposed to take over and discuss prospective trials in a different setting where now we don't know the effective treatment and we want to test experimental treatments. Needless to say, this is a more complicated situation. The first type of design that can be used in this situation, in fact, is a very sort of straightforward design. We now have an experimental treatment. We think we know the predictive factor that predicts the effective treatment, and therefore in phase 2 or in phase 3 we can use a targeted design. We can in fact select the patients according to the predictive factor that we believe is well known. For example, Herceptin trials in advanced breast cancer. These trials were targeted at patients who had HER2/neu overexpression, and so these designs in fact excluded patients who were biomarker negative. That means patients who did not have HER2/neu overexpression. These designs do not pose a particular problem. If we wanted to do a randomized design, same thing. We would actually select patients based on their biomarker value, in our example HER2/neu positive patients, and randomize these patients between Herceptin and control. So these designs are very straightforward. In phase 3, likewise, targeted designs are very straightforward. An example of such a design is the TOGA trial in advanced gastric cancer. This design is interesting because it's the same molecular target as in the advanced breast cancer example. It's HER2/neu overexpression. And so here patients were selected, patients with advanced gastric cancer were selected based on the HER2/neu positivity, whether they had HER2/neu amplification or not. Patients who were amplified indeed were randomized between chemo alone or chemo plus Herceptin, and patients who did not amplify overexpress HER2/neu were actually excluded from the trial. So the trial targeted patients in whom the benefit of trastuzumab was thought to exist. And indeed, here are the results of this trial. These results were shown about a year ago at ASCO, and you see that indeed there was a large benefit of Herceptin in that subset of patients who were HER2/neu amplified. Very significant p-value and a hazard ratio in favor of Herceptin of 0.74. Again, the benefit of these targeted trials is that patients who are entered in the trial are thought to benefit from the treatment, and indeed the benefit can be quite large. But there are some issues with these designs as well. Targeted designs may be less efficient than randomized-all if the drug has at least some activity in biomarker-negative patients. And talking about the effect of trastuzumab in breast cancer, even though trastuzumab was shown not to have response rates in advanced breast cancer patients, in fact there is still a question, an open question, as to whether trastuzumab may have a benefit in HER2 non-amplified patients in the adjuvant setting. So the question of the effect of trastuzumab in biomarker-negative patients, in other words, in patients who do not amplify HER2/neu, in fact that is still an open question today. So doing a targeted design may represent a loss of opportunity for biomarker-negative patients, and in fact another advantage if the biomarker is poorly known is that in a randomized-all design multiple candidate biomarkers can be tested, whereas in a targeted design, in a sense, you are stuck with the biomarker that you thought you knew by definition. Now let's go to a more interesting phase of development, the phase 2. Again, we want to develop a new treatment, and we think we may know a biomarker, but we're not completely sure that this biomarker is predictive. If we were sure, we could do a targeted design, but now we're not quite sure. So one design we could use in those situations is the so-called adaptive phase 2 parallel design. And the example here is dovitinib in breast cancer patients. Here the design consists of doing in fact two stages designs in parallel. So in fact a biomarker negative two-stage design and a biomarker positive two-stage design. So the patients are stratified by the biomarker positivity, and then in each of the two strata a two-stage design is carried out. After the first stage patients are entered, the drug, the new drug under treatment, under investigation, may be active or inactive. If the drug is found to be active, then that stratum continues to stage 2, and likewise for the other stratum. And if the drug is found to be inactive, then that stratum is stopped. So this, in fact, consists of doing two stratified, two-stage designs in parallel using stratification for the biomarker. An example of this was, again, in breast cancer. It's a phase 2 clinical trial of a drug called dovitinib, monotherapy in patients that are HER2 negative now, patients who have metastatic breast cancer, HER2 non-amplified. And again, this design consists of doing two parallel two-stage phase 2 designs, one in the FGFR1 amplified patients, and those patients were presumed to benefit, and the other one in the FGFR1 non-amplified patients who were thought not to benefit. And again, the trial proceeds to stage 2 if all patients benefit, but it doesn't proceed to stage 2 if indeed one of the two strata doesn't show enough activity. Another type of design that can be used in the same situation, phase 2, with an experimental treatment where the biomarker is not known at all, so here we don't even know what biomarker to use, and therefore we cannot stratify for it, is to do a tandem two-stage design, an example of which is saracatinib in pancreatic cancer. So here the design is different. It starts with a two-stage design in all comers. If indeed in all comers the drug has enough activity, the second stage proceeds. But if after the first stage the drug is not found to have enough activity to proceed in all patients, a biomarker is identified in all patients, and the trial is continued only in the biomarker-positive patients. So this design, in fact, allows you to test an experimental treatment and at the same time identify a biomarker that predicts the effect of this treatment. And the second stage design will only continue in the biomarker-positive patients. An example of this design, again, is in pancreatic cancer. Patients who are treated in this phase 2 clinical trial of saracatinib as monotherapy in previously treated patients, or refractory patients with metastatic pancreatic cancer. The primary endpoint is the 6-month overall survival, and in this trial, after the first stage, in fact, the trial failed overall. That drug did not have enough activity in all comers to continue to the second stage. So the second stage continued only in patients identified as a potential responder by a pharmacogenomic biomarker that was developed on the first stage patients. So this is a potentially quite interesting design, but it has its problems as well, and one of the problems is the sample size. If we want to completely validate a putative biomarker, we must do this in a phase 3 trial, not in a phase 2. So the phase 2 trial is really a screening phase to identify a potential biomarker or putative biomarker, and then the phase 3 is required to validate the true predictive value of that putative biomarker. So two designs will be discussed in this section, so-called enrichment designs and the prospective subset design. Again, we're back to phase 3 now, relatively large numbers of patients, and these are confirmatory trials. A first example of such a confirmatory trial is called the IPASS trial. This was a trial of IRESA, or gefitinib, versus chemotherapy in patients with non-small cell lung cancer. And this trial was actually performed in Asia. The reason they performed this trial in Asia was that, in fact, in early phase 2 trials of gefitinib, it was found that gefitinib had a very high response rate in Asian patients and did not have a high response rate in other patients. So it was thought that Asian patients had some biomarker that predicted the effect of gefitinib. And it was later found, not when the IPASS trial started, but after the trial started, that in fact EGFR mutation explained the benefit of gefitinib as compared with chemotherapy. If you look at the results of the IPASS trial that were published in the New England Journal of Medicine, it's obvious that gefitinib has a big benefit compared to chemotherapy in patients who have the EGFR mutation. Again, this was not known before the trial started. All that was known was that Asian patients seemed to respond better to gefitinib than non-Asian patients. So the trial was enriched in GFR mutation, but this was known only after the fact. Patients who did not have the EGFR mutation, in fact, did not benefit from gefitinib at all. On the contrary, these patients in fact had a worse outcome than patients who received conventional chemotherapy. So here we see very clearly that EGFR mutation is a predictive biomarker for the benefit of ERESA. Patients who are EGFR mutated have a big benefit of ERESA. Patients who are not EGFR mutation actually do not benefit from ERESA, and still worse, they fail to benefit from the chemo that they would otherwise have received. So putting everything together, this is the curve as published in the New England Journal of Medicine. This is all patients combined, whether they have or don't have the EGFR mutation. And what we see here is a very confusing type of figure where the two survival curves or progression-free survival curves cross each other. The gefitinib curve is initially lower and then it becomes better than the chemotherapy survival curve or progression-free survival curve. And this is simply because these curves arise from a mixture of two distributions, the distribution in patients who are EGFR mutated and the distribution in patients who are not EGFR mutated. So these overall curves, in fact, are not very informative, and the hazard ratio here doesn't mean anything because we have non-proportional hazards over time. So these curves have to be interpreted with great caution, and it's very fortunate that the EGFR mutation is now known, and it does explain why results differ depending on this biomarker value. Another approach, and this will be the final design we discussed today, another approach to confirm or to validate a predictive biomarker is to use a prospective subset approach. And here, in fact, the idea is to test the hypothesis overall in all patients, and also to test the hypothesis in the biomarker positive subset in whom we believe that there will be a larger treatment effect. So we will do two comparisons at the end of the trial. First, an overall comparison ignoring the biomarker, and second, a comparison only in the biomarker positive patients. So we will have to split the alpha level in two parts, an overall part and a biomarker positive part. And if 5% is used overall, we will have to allocate a portion of this 5% on the overall hypothesis and another part of this alpha level or significance level on the biomarker positive subset. Now, of course, there are many ways of doing this allocation of alpha between two different hypotheses. The easiest way to do it is to simply split the alpha and say if I have 5% here, I can take, for example, 3% overall and 2% in the biomarker positive subset. But of course, this is not a very smart way of doing it, and there are better ways of doing this while still controlling the overall alpha level. And these different methods, which are referenced at the bottom of the slide here, account for the fact that the two tests are correlated. Obviously, the biomarker positive test is a subset of the overall test, and we can make use of this statistically to gain power by allocating the alpha in an optimal fashion. An example of this design is the SATURN design. This trial actually randomized patients who were chemo-naive with advanced non-small cell lung cancer. All these patients had a tumor sample taken, and after 4 cycles of a standard chemotherapy, they were then randomized, if they were not progressive disease, randomized between maintenance treatment with erlotinib versus no maintenance treatment, placebo. And this randomization was stratified for all of these factors. Importantly, it was stratified for the EGFR positive or negative, and this was thought to be a predictive factor. So the hypothesis of interest here was that indeed erlotinib was an effective maintenance therapy in all patients. The alpha level used for that hypothesis test was 3%, and also that in fact erlotinib was a significant or was an effective treatment for patients who were EGFR positive, and this was tested at an alpha level of 2%. You notice here that there was no adjustment for the fact that the two tests are correlated. An alpha level of 2% was spent here, and an alpha level of 3% was spent there. So the sum of these two is still 5%, but obviously this is not an optimal strategy, so it's a conservative strategy. There were secondary endpoints also in this trial as listed here. So here are the results very briefly of this trial, and the references are listed here at the bottom. In fact, it was found after the trial that the EGFR positivity was not at all predictive, contrary to the expectation, was not at all predictive of the outcome. As you can see, the 12-week progression-free survival and the 24-week progression-free survival was almost identical in the overall group, as it was in the EGFR IHC positive group. As you see, these proportions of patients who are progression-free at 12 and 24 weeks are almost identical, and then, of course, not surprisingly, the hazard ratio was almost the same in the positive biomarker group as well as in the overall population. It was, by the way, highly significant in both, showing the benefit of erlotinib as a maintenance treatment in these patients. So this concludes the different designs I wanted to look after and to present, but there are some key questions to choose between these designs, and the key questions are listed here. First of all, what do we know reliably before the trial starts? If we knew a lot about the biomarker, then we could use them quite effectively to target the treatment at those patients who are thought to benefit. The reality is that very often we don't know much about the biomarker, or what we know is not confirmed or is not fully established. So we need to then, confirm that the biomarker indeed has the effect that we think. Secondly, what do we want to know reliably after the end of the trial? Some of the phase 2 trial designs that I presented and discussed are very smart designs, but they will not provide a definitive answer, and after the trial is over, we will probably have to do phase 3 trials to confirm the discovery that we have hopefully made in the phase 2 design. Thirdly, what are the consequences of type 1 and type 2 errors? In other words, what is the consequence of having a false positive biomarker effect or a false negative biomarker effect? Is that important? And more importantly still, are we able to ever repeat a trial after a trial has been done if we find a positive or a negative answer? So all of these questions are important, and they will drive our decision to choose one design rather than another. And finally, I wanted to finish this talk with a quote, and this is a quote by Aristotle, so it goes back quite a while, on personalized medicine. So personalized medicine, of course, is the theme of the day, but it's amazing to see that it goes back quite a long time. And this is what Aristotle had to say about personalized medicine, and I personally believe that it remains valid today. Obviously, we would love to predict the outcomes of individual patients, but that, I think, at the moment is completely science fiction. So what we want to do before we can go further in the direction of adjusting treatments to patients is to not do individualized medicine, but validate biomarkers. And that, I think, will pave the way to reliable biomarker discovery. So with that, I will close. Thank you for your attention. And again, as Beverly said to begin with, we would have loved to take questions during the webinar, but as I was alone today, you will realize that it would be very difficult for me to handle questions in real time. So instead of which, if you have questions, please submit them by email, and then we will try to respond to your questions individually. Thank you very much for joining the webinar, and hope to see you or to have you present on the next one, which will be early in 2011. Thank you very much, and goodbye.